First of all, thank everybody for being here, and unfortunately I am not going to speak in Finnish. I'm Italian, and I moved in Finland for my doctoral studies like 3 years ago, and since then we have been trying to find new therapies for cancer. And why would we do that? The problem with current therapies is that we have a lot of compounds like cisplatin, drugs like doxorubicin, and also therapies like radiation therapies. And the problem with those is that they are extremely good at killing cancer cells, and the way that they do it is simply by damaging the instructions, as before Anna was telling us. Every cell has an instruction book from where it reads how to make proteins. Once this instruction book is broken for some reason, then the cell dies because the cell doesn't know how to live anymore. And this has been the aim of the research in the last 30 years. But the problem with that is that these drugs have also some side effects, so they also harm normal tissues. Therefore, we looked for ways of killing in a specific way cancer cells and not make any danger to normal tissues. And one nice way to do that is to use viruses. And it might be like weird to use something dangerous as viruses to kill cancer. And I think many of us have seen some films where there was a huge apocalyptic scenario where zombies were killing everyone and they were coming from some crazy viruses that the scientists in the lab manipulated. So far I didn't make any zombie yet, but maybe by the end of my PhD I can make one. Let's see. But the thing is, we now know with all the genetic techniques that we have how to safely engineer viruses and make them specific. And I'm particularly proud of bringing to your attention the fact that since the start of last century, some relationship between virus infection and cancer growth has been noted by some clinicians, in particular by an Italian one here, De Pace. And he had this patient with huge cervical cancer. And then at some point, the cancer disappeared. The patient didn't undergo any surgery or other special treatment, just that the cancer at some point was not there anymore. But this clinician and many others after him started to notice that when patients with cancer get some kind of infection, it might be measles infection, or even if they get some vaccination with viruses, their cancer will there start to regress. And therefore, it was noticed, this link between viral infection and cancer regression. And even nowadays, there was a nice episode from Dr. House where he had this patient, and the patient had cancer, but this patient won't take any cure because she was very religious. She was praying God to cure her cancer, and then she didn't receive any treatment. And then at some point the cancer disappeared. And everybody was saying, okay, this is the will of God. I was praying so much and I got cured. But then House realized that while this patient was in the hospital, she had an infection from herpes virus, and this infection cured the patient. So when I saw this episode, I was like, wow, this is not possible, because I was still doing my master thesis, but then I knew that there were actually some viruses who could do that. And people in the last century has been studying that, and only now, in 2015, after 100 years of learning, we had the approval of the first drug based on viruses. It's an herpes simplex virus. Amgen has have it approved by FDA in the US, and it's for the treatment of melanoma. So it took a long time for us to understand and exploit this mechanism. But unfortunately, these viruses by themselves cannot do much. We need an extra boost, an extra help. And in our research group, we think that if we combine these viruses, which can kill cancer cells, but if we combine them with the power of our own immune system, then this therapy can improve. And why we think that the immune system is important? Because if you think about it, the cancer, every kind of cancer like melanoma, it comes from normal skin. As well as lung cancer, it comes from our own lungs. So there must be something wrong about the control and the surveillance that our immune system makes in our body. Because it's like very difficult to think that the immune system can let cancer cells escape from its control. And in recent years, there has been a huge revolution in cancer, and in particular in the oncology field, where people have started to understand that cancer is not really about some group of cells which are proliferating and growing without control. Cancer is about a group of cells that can hide themselves from our own immune system so that they don't get killed. And if we can think about this as a way to exploit this mechanism, if we can teach to our own body to find the cancer cells and kill the cancer cells, then all of a sudden we will need much less chemotherapy, we will need much less radiotherapy, because our body will take care not only of the primary tumors, but most importantly about all the metastases that we cannot reach with the drugs. For this reason, combining the immune system with the viruses that can kill cancer was our aim. And what we did is to build a platform which we call PeptiCrad that has two kind of mode of actions. On one side, we have a virus which can kill cancer cells, but on the other side, our virus can activate the immune system and teach to the immune system what a tumor cell looks like, and eventually the immune system will clear the other cancer cells. And the way that we have done it is basically by taking a normal virus. It's in our case an adenovirus, so it's the virus that is causing like throat pain or cold or cough. And we covered this virus with little pieces of tumor, so that when our immune system sees the virus, it's also seeing tumor peptides or tumor pieces, and then the immune system knows what it has to kill, what it has to find. And we have been able to validate this system in different tumor models, and I will just show you some data that we obtained by studying mice, which is the in vivo model that we use. So, just quickly, here, what you can see, we have mice, and we treated mice with different compounds. Some mice got just water, which is just the placebo, and in fact, their tumors were pretty big. Then some other group of mice, they get the normal virus, which in our case didn't seem to have any effect on this very aggressive melanoma model. But then when mice were treated with our platform, you can see that the volume of the tumor was like half of the other controls. So in our case, we had some kind of synergy between these viruses and the immune system. And what I would like to point out is that these volumes of the tumor are actually tumors which were not directly treated. This means that our platform can not only have an effect on the tumors that you can inject directly, but also eventually on metastases that you cannot reach by the treatment. And this was the case. And then I would like to show you another example, because so far this is done in mice with mice immunosystem, murine tumor cells, so everything from a mouse. But then we want to see, can this be of any value for actually humans? So can we try this in a more challenging model? Of course, we couldn't like take people and treat people. We don't have the power and the resources to do that. But we have another model which is called humanized mice. It's basically some mice where you have a human immunosystem engrafted into the mouse, and you can give to the mouse human tumor cells. So you have some kind of human replica that you can use for your studies. And it's a very good model. And here you just have the time of our experiment, which lasted 17 days. And all the mice started with some tumors. And then you have some mice with the, let's say, dashed line, which just got the placebo, which is the water. And you can see that at the end of the experiment, their tumors were just growing until this size. And then in another group of mice, we have treated their tumors with the normal adenovirus. And you can see that the tumor kind of stabilize and then they regress a little bit, which is a good result. And it's results that we are seeing in the clinic every day. Like when we give these viruses, most of the tumor of the patient, they experience some kind of stabilization. But when we treated the mice with our platform, then you can see that the tumors quickly disappeared until day 17, meaning that when you combine the viruses with the power of our own immunosystem, or their own immunosystem in case of mice, you might achieve tumor regression. This is very, of course, nice results for us, and it does not mean anything yet for humans, because we need to take this to the next step, which is human clinical trials. But so far, we are trying to establish a company and raise some funding to make a small clinical trial with real patients. And we hope that it will be, of course, as successful as our data here. And with these, I would like to conclude. And of course, I thank everybody here. And you had actually the patience to bear with me during my presentation, but also all the colleagues and friends, more than colleagues, and especially the supervisor. And if you have any questions, you can ask me now, or you can reach me at my email. I will be very happy to chat and discuss or whatever comes to your mind. Thank you very much.